first topic is drug discovery and development, which is like as like till now you have come to know that throughout the clinical research procedure, pharmacovigilance, and clinical data management also, for that, for all those processes, there is a need of some substance that is to be tested and their data has to be collected and their data has to be managed. So what exactly is that substance or the thing? Just we are doing everything. Like we are doing the research, we are maintaining the data, we are doing the pharmacovigilance, everything. That is for the drug. So how drug is discovered and how it is developed is what is our first topic in the first module. So yeah. So okay, if we look at the definition of drug, I would like you guys to tell me what according to you is a drug. Explain in your own simple terms. Sakshi, what according to you is a
Can I?
drug? Yeah. Yeah, you can.
Drug is an active substance that is used for treatment, diagnosis, or a cure.
Okay. Okay. So yeah. So drug is any substance which could be a chemical or biological, which is used to diagnose, cure, mitigate, treatment, or prevention of disease in humans or other animals, and to affect the structure and any function of the body or human or of human or other animals. So which means a drug is any substance, whether it is made in lab, which is like chemically produced, or produced naturally, which is like biologically produced, that can find, like diagnose means to find, cure means treat, mitigate means you are having pain, but you are reducing the pain like that. So mitigation means the disease itself is not cured completely, but the symptoms or the side effects or the pain or anything, it is being lowered. So that mitigation or to prevent a disease. It can also change how the body works. Like sometimes we take supplements not because of some disease, but we want to boost our immunity or like that, or either to make it function better or to just to fix a problem like indigestion or something that is not a disease, but better function of our body. So as we all know, paracetamol is a chemical drug which is used to treat fever and pain. So and insulin, which is like biologically produced, so it is a biological drug used to treat diabetes, which controls the blood sugar level. So see, some like something which is chemically derived as well as biologically derived, but it is used to cure, diagnose, treat, mitigate some of the other disease or some problem, or to make our body function better, then it is termed as drug. If we say drug discovery, which means identification of a new chemical entity as a potential therapeutic agent. For example, if we say many of our, like not many, almost everybody knows that having turmeric into milk boost our immunity, help to heal us more quickly. But the whole turmeric itself is not the active compound. The active compound is the curcumin, or some of the active part is there in the substance that actually works for that substance, that actually work for the disease. So so if we look at the term called drug discovery, then we have to look at the potential active ingredient which can be converted or which can be used directly or solely into a patient or into an individual for its better effect. So, like drug discovery is the first step. So, scientists research for a new molecular compound that could work as medicine. This involves studying how disease work and finding substance that can target or block the problem inside the body and everywhere. For example, think of like searching for a key that fits a lock. Lock is disease target and key is a new molecule. For example, in COVID-19. So COVID-19 was a disease. So scientists discovered a medicine called remdesivir. It was very much famous and everybody know about that medicine like as far now. A compound that could stop the virus from multiplying. So that's called a drug discovery. The third point comes is drug development. So it's like we came to know that the active ingredient in the turmeric is curcumin, but we directly cannot give curcumin as it is to some patient or to an individual. It has to be either in a form of syrup or in a form of supplement or in a form of medicine. So it's like a development of that compound, which simply means once the potential drug is found, it doesn't immediately reach patient. It must go through multiple stages to check if it's safe and effective, what could be the dose. This includes the preclinical testing in animals, clinical trial in humans, and submitting regulatory approval application to agencies. So preclinical, what do you mean by preclinical testing? Sakshi, what do we mean by preclinical? Like, what does this term means?
प्रीक्लिनिकल मीन्स ड्रग मार्केट में अप्रूव होने के पहले जो उस पे ट्रायल्स जो करते जो अभी मैंने एक्सप्लेन किया.
Okay, but why it is called as preclinical? So it could have been called clinical trials only if it is being tested. But preclinical exactly means what? It is tested on whom? Okay, I'll make it more
ऑन
simple. Is it
एनिमल्स.
tested on humans? Okay, it is tested on animals. Is it tested on humans? Yes or a no? Is it tested on humans in preclinical phase?
नो इट इज टेस्टेड ऑन एनिमल.
Perfect. Okay, so preclinical means clinical itself means testing on humans. So anything which is done prior to testing it on humans is called as preclinical. Whether it is like uh, whether it is like you have found some flower in the garden and you know it could have some active compounds. So you
यस
plug
यस.
the flowers, you have taken it into the lab, you tried its active compound, you tested on microorganisms, you tested on bacteria, on anything except humans, it is called as preclinical thing. So it is preclinically tested, which is on animals to see if it works and is safe or not. Because if it's not safe for microorganism as well as for the animals, then it cannot be taken forward into humans. Then comes the clinical trial, which means testing on humans in three main phases: phase one, phase two, phase three, and phase four is like a post-marketing surveillance. So three main phases to confirm its safety and its dosage,
इस.
like at what dosage it is safe and it is effective as well, and the effectiveness. Like we don't want to give a higher dose of any of the compound because sometimes it is like even if it's a good compound, but like if it is taken, if it is not taken appropriately, then it can harm. So safety, dosage, and effectiveness, all of these are tested into the clinical trial phase. Then there comes the regulatory approval, which means all the data is submitted to government agencies. So there are different government agencies specific to India, DCGI, FDA
यस.
for USA,
एफडीए.
EMA for Europe. Yeah, to get permissions for public use. So, for example, paracetamol went through animal studies first, then human trial to test the dosage and safety, and finally got approval before being sold in pharmacies. Similarly, for COVID-19 vaccines also, they went through rapid development from discovery of the viral target because that was an pandemic, so it was spreading a lot. So there was very much what to say, a rapid development. Then from discovery of the viral target to human trials and then regulatory approval, everything was done. But without approval, it was not taken out into the market. So all the regulatory approvals were done before we could get uh, those shots at the vaccination center. So do you have any doubt in these terms? So we have understood what is drug, what is drug development, and what is drug discovery.
यस.
Okay. Can we move to next slide? Okay, so the thing was, we start from drug discovery. We came to know that during the drug discovery there can be many compounds that we find, that we feel that they have the potential to be that one specific medicine which can work for that one specific disease. So currently we don't know, we are just searching. We have gone into labs, we made something, we think okay, this can also work, this can also work. So we have around 10,000 compounds. These are just number, we are not very sure, these are just for example. Then while we test it during in the lab with all our microorganism as well as in animals, slowly we come to know that out of those 10,000 compounds, only 250 could be tested properly, and out of that 250 compounds, only 5 compounds are such which really have the potential, and they are safe also. Not just having potential is a checkbox to be ticked, but it has to be safe first of all. So some out of 10,000, only 5 compounds you found effective, as well as um, they were effective as well as safe. So only those 5 compounds out of those 10,000 will be going into clinical trial phase. In clinical trial phase, you will be testing those compound onto real human beings, like alive people. There would be no microorganism, no very much controlled space. They would be patients as well as healthy volunteers as well. And out of those 5 compounds, if you find some or the effective dosage or um effective medicinal view, then you'll be submitting all your documents for the regulatory authorities for them to review that. After the review is done, and if every of your document as well as the process you have been performing is regulated and according to the SOP and according to all the rules and regulation, then only the regulatory authorities will approve your drug or approve the compound, and that will be sent out into the market for sale. Otherwise, if you have not regulated your the process properly, as well as no compliances are being maintained, then the FDA will directly cancel your trials, and it would not allow you to launch your product into the market. So into the second diagram, we see what exactly, like how much time it takes. So we can see it takes minimum around like 10 to 20 years also for a drug to come into the market. So it's a very long process. And if in between the regulatory authorities are not satisfied with all your trial procedures and everything, so you can understand there's a big loss for the companies for the sponsors who are conducting these trials, because they have to give a lot of money as well as a lot of time into this process. So if we bifurcate the phases, so we know first comes the preclinical phase, then clinical phase, which consists of mainly phase 1, phase 2, phase 3. In preclinical phase, we know there is a laboratory and animal study, and the purpose of this is safety and biological activity to exactly see because we just hypothesized it, that the compounds, that those 10,000 compounds will work, but are they actually working or not? Are they having little bit of activity also or not? All these things are being tested into the preclinical phase itself. Course time is around 1 to 2 years. It can extend, it can be lowered. So these are not just fixed numbers. It could vary accordingly. And so, like this, in the preclinical phases, we look after the safety and the biological activity. Then we file for IND. IND is investigational new drug. So, investigational new drug means it is still in the investigation phase. A new drug is not been launched as drug right now because we are still studying it. So, it is called as IND, investigational new drug. After filing a IND to the authorities, so it is like we are submitting all our documents which we have collected in the preclinical phases to some authority, regulatory authority. They will go through everything, that whether we have completed and we have completed and maintained the decorum of the whole preclinical phase. We have not manipulated the data, and it is safe to be used into the human. Then only they'll accept it and they'll give us the permission to use that compound or those compound which have been selected into the preclinical phase into the clinical phase onto humans. So in clinical phase, there is phase one in which we conduct the trial onto 20 to 100 volunteers. So, and in that we mostly look for safety and dosage, because now we have came to know that, oh, okay, those five compounds are those were working good in preclinical phases, but are they safe in humans? And if they are safe, at what dosage they are safe? This phase is mostly for 3 to 4 years. And once in phase one, we come to know, okay, that it is safe and at what dosage. For example, we try giving it at 1 mg. 1 mg, it was not showing any effect. It was not showing any effect. It was not showing any side effect as well. 2 mg, it started showing a little bit of effectiveness, but without any side effect. Then we try to give 3 mg. At 3 mg, we came to know that it is showing effect, but it is also showing a lot of side effects. So like that, we will come to know what exactly the dosage is to be given to the patients, so that there is effectiveness. Even though effectiveness would be less, that would be fine, but there should not be any side effect or any harmful reaction. So that is the primary thing that is kept in mind. Then in next phase, phase two, we increase the number of patients that goes from 100 to 300 patient, and we look after the effectiveness and side effects. So in phase one, we came to know that which dose is safe and which dosage is safe. And in phase two, we see that dosage is safe for how long, how much effective that dosage is. Like for example, you are giving that 2.5 mg for a week. Then you have seen that after giving it for a month only, you are seeing some effectiveness if you are giving it to 2.5 mg or something. So you'll increase that mg and you will see at what dosage the maximum effectiveness is there without having any side effects. That takes a lot more time, 4 to 5 years. This is also like very much a approx time period. Then in phase three, we again increase the number of participants. It goes from 300 to 3,000 patients. So the patient ranges from 1,000 to 3,000 patients. And the purpose of this phase is to verify the effectiveness and long-term side effects. And this is the longest phase out of these all three. So for 6 to 8 years or 10 years or 12 years, that totally depend upon the compound or the substance we are looking into. So it verifies the effectiveness. So like we have understood at what dosage the are active, or the our IND is effective and it is not showing side effect, but for how long it is not showing side effect. Because uh, after a time, what happens, our body becomes like our body adapts the condition, our body becomes very in a way like it doesn't react to anything, so it doesn't show effectiveness, or it starts showing side effects after a very long time. So that also we have to make sure that if some medicine has to be taken for more than a month or 2 months or 6 months, then it could happen that in starting it was not showing any side effect, it was very effective, but as the time period goes on and the time period passes, it loses its effectiveness and starts showing the side effects. So that also has to be kept in mind. So once the phase three is complete and we know that, okay, for this, like for a year, we tried giving that same medicine and same dosage, and there was the same effectiveness and there was no side effects, very minimal side effect. So after that, if such things comes, then the phase three is being passed, and then all the documents again are being collected and filed for NDA.